The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
This is the second recitation on cholesterol, and it's really sort of focused on this question of how do you sense cholesterol in a membrane. Okay, so that's really sort of a tough problem. They've developed new tools, and that's what we're going to be talking about, what the tools are and whether you would think they were adequate to be able to address this question about what kinds of changes in concentration of cholesterol, number one, can you measure them? And number two, what effects do they have in terms of whether you're going to turn on cholesterol biosynthesis and uptake because you need more cholesterol, or you're going to turn the whole thing off. Okay, so we've been focusing, as we've described in the last few lectures, in the endoplasmic reticulum. And what were the cholesterol what kinds of changes in cholesterols did they see in the experiments they were doing in this paper? What were the range of changes that they saw?
3 to 10%?
Yeah, so I don't see, so something low. Say they were trying to do this same experiment in the plasma membrane. How do we know it's the ER membrane that does this sensing? That's what the whole paper is focused on. That's what everything we focused on in class. Say you wanted to do a similar kind of experiment in the plasma membrane. Do you remember what I said about the levels of cholesterol? So they're distributed throughout the cell, right, in all membranes. Where is the most cholesterol? Okay, so if you don't remember, it's the plasma membrane. Okay, so say instead of having 7 or 8% of the lipids cholesterol, say you had 40%. That's an over-exaggeration. Do you think this kind of an experiment would be hard to do that they've talked about in this paper? So you would want to do this if you tried to do the same experiment with the plasma membrane. So the key issue that you need to think about is go back and look at the changes. You know, they did a whole bunch of different experiments. They sort of, I mean, the numbers are squishy, but they came up with numbers that sort of reproduce themselves, I thought, in a sort of an amazing way. But now say you wanted to do this in the plasma membrane where the levels of cholesterol are much higher. Do you think that would be easy to do? Using the same techniques that are described that we're going to discuss, or not? And what would the issues be? Yeah?
So they had to deplete the cholesterol from the membrane, and so that would it would probably be hard to deplete it to a level that's low enough so that you don't get the activity, right?
So I don't know. So that's an interesting question. So you'd have to deplete. So that's going to be it. We're going to have to control the cholesterol levels. But what change, if you looked at the changes in levels of cholesterol in the ER, how much did they change? They changed from what to what? From 2% to 7%, okay? Say that you were sort of in that same range of change that was going to turn on a switch in the plasma membrane, and say you could control the levels. Do you think it would be easy to see that? So you start with 40%, say. That's sort of the norm. Say the change was very similar to what you see in the change in the ER. Do you think that would be easy to detect? No, because now you have two big numbers, and there's a huge amount of error in this method of analysis. So those are the kinds of things I'm trying to get you to think about. I don't know why it's the ER. I mean, everybody's focused on the ER, okay? Could cholesterol and other organelles have a different regulatory mechanism or somehow be connected still to what's going on in the ER? It could be. I mean, we have the simplest, you start out with the simplest model you can get and you test it. But then as you learn more, or people, you know, we have more and more technology, we learn new things, you go back and you revisit and rethink about what's going on. So the key question is, it's really sort of this switch of having cholesterol that keeps it in the membrane, or not having cholesterol. And the question is, what are the differences in the levels that allow turn on of cholesterol biosynthesis and LDL biosynthesis, which then allows uptake of cholesterol from the diet. Okay, so that's the question. And what does this look like if people hadn't measured this by any method? And this model I've gone through a number of times in class today, so I'm not going to go through it again. Hopefully you all know that in some form in your head, or you have the picture in front of you so you can remember it. So these are the questions I want to pose, and I want you guys to do the talking today. Okay, and what I'm going to do is I have most of the figures on my PowerPoint, so we can bring them up and look at them. And you can tell me what you see, and then everybody might be seeing something different. And so we're thinking about this differently, and maybe we come to some kind of consensus about whether these experiments were carried out well or not. So one of the first things, so these are sort of the general things, and then we'll step through them. But they wanted to perturb the cellular cholesterol levels. And how did they end up doing that? Did that make sense? We talked a little bit about this already. I mean, what did they use as tools to do that? So you need to speak louder, because I really am deaf, sorry.
So just, they were careful about the amount of cholesterol present in the media.
So that's one place, so they can deplete cholesterol for the media, but then what do they do? So the whole paper is sort of about this. How do they control the, so let's assume that they can do that, and that's, you know, they got good at that. I think a lot of people have used that method, and so they can deplete media. So how did they deplete cholesterol? There were some unusual ways to deplete cholesterol in this paper. Did any of you pick up on that?
A chemical that can bind to cholesterol.
Right. So did you think that was unusual? Did any of you look up what that was?
There's a kind of carbohydrate, right, and can bind to cholesterol.
Yeah. So, but what was interesting about it, it was, you know, hydroxypropyl. Remember HP, cyclodextrin. Okay, we're gonna look at this in a minute. But what do we know? What was the other molecule they used to add cholesterol back?
Another form of that molecule is MD.
Right. So methyl cyclodextrin. I'm going to show you the structure, but they aren't very different. So, I mean, have any of you ever heard of cyclodextrin before? I mean, people won the Nobel Prize for that. Don Graham won it. Breslow spent his whole life studying host-guest interactions. So, I mean, you guys, I don't know what they teach you now anymore, but I mean, that used to be something that was taught a lot, host-guest interactions, trying to understand weak non-covalent interactions as the basis for understanding catalysis. But to me, that was immediately when I saw this, what the heck's going on? So then I Googled it, and, you know, immediately, you know, I don't know anything about hydroxypropyl. You Google it, you look it up, and then you look at it, and, you know, if you were a chemist and you were really interested in the molecular interactions, you might make a model of it and then see if you could, you know, what is the difference between that one little group. When you look at the structure, it's sort of amazing. And that's like the basis of most of the experiments. So you need to believe that they figured that out, okay? And that's not in this paper. So if you really cared about it, you would have to go back and read earlier papers and see what are the experiments that led them to focus on these molecules. How else did they end up getting cholesterol levels back into the cell? Do you remember what the other method was? So we'll come back and we'll talk about this in a minute. So that was one of the methods.
They added two kinds of sterols.
Okay, so they did add two kinds of sterols, and they're trying to figure out, this is sort of another unknown. What was the difference between the sterols? Simply a hydroxyl group. Okay, so you had, if you looked at this, cholesterol is this guy. Okay, and then they had something like this guy, 25, I don't remember where is this out in the side chain, hanging out of the little shear system you have. I don't think they learned very much from that. And in fact, on your problem set, you had all of these different cholesterol analogs. I mean, I think we still really don't get it. I mean, that's complicated. We talked about this in class. You have these transmembrane helices. What is it that's actually the signaling agent? So people are still asking that questions, and we haven't quite gotten that far. But if you've read the reading for HMG-CoA reductase degradation, which is what we're going to be talking about in class, the signaler is not the sterol, it's lanosterol. Okay, now where have you seen lanosterol? The biosynthetic pathway sort of has lanosterol sitting in the middle. It's not all that different structurally from cholesterol. You need to go back in. It's got four. They both have four-membered rings. They have different extra methyl groups. So people are trying to sort of sort that out. I don't think we really know. But how else? So they had. So you're right. They used sterols. They didn't use that. They didn't see very much difference with the sterols. What was the other way, which is sort of unusual, that they added cholesterol back into their system? So they could add it back with the methyl cyclodextrin. They told you that that worked, and if you believe that, then, and you look at the data, it looked like that was happening. Nobody remembers? Okay, well, we'll get to that in a little bit. Okay, so the question we're focusing on is, what are the changes in concentrations of cholesterol in the ER? Okay, so what method did they use to try to separate the ER membranes from all the other membranes?
They first separated the pellets, and then they did a...
They separated the what?
The unlicensed cells and the nucleus in the pellets.
Okay, so that's good. You can separate out the nucleus, and you can do that by ultracentrifugation. We've seen that used, a number of different kinds of ultracentrifugation. We've seen that the different particles, that the lipoproteins in the diet, how do we separate those? We talked about that in class briefly. You haven't had any papers to read, but what was the method of separation? If you look at all those particles, remember we had a little cartoon of all the particles, and we focused on LDL, which is the particle that has the most cholesterol, so that's why everybody's focusing on that. What was the basis of the separation? Was the what? You need to...
Did they use a sucrose gradient? Like ultracentrifugation?
Yeah, ultracentrifugation, but what was it? How did...
With a sucrose gradient?
Oh, okay, yeah, okay, so they have different density gradients, okay? And so that's going to be a key thing, and that's because if you look at the composition, they have different amounts of proteins, different amounts of fats, and they have they float differently. So that's the method that they're going to use here. Is that a good method? Can you think of a better method? So in order to sort of understand the switch for cholesterol, you got to be able to measure the changes in cholesterol, right? Not an easy problem, because cholesterol is really insoluble in everything, okay? And so how much is really in there, and how does it change under different sets of conditions? So is this a good method? What do you think? We'll look at the method in a little more detail when I pull up the figures, but what did you think when you read the paper?
I think it's a pretty good method, because other than their slightly different densities, there aren't any other like properties different from the membrane, say plasma membrane and Golgi bodies and ER. So it's like the only way I could think of.
Yeah, so the question is, could you separate? I mean, even separating the nucleus from the cytosol is not so trivial. Okay, by these methods, these are really gross methods, and during the centrifugations, things diffuse. So if you're having close separations, it's equilibrating down this thing, and so you're getting your proteins or your lipids are spreading out. Is there anything else any of you experience with insoluble? I mean, this is what we're dealing with, is an insoluble mess And how do you separate things in a way that you have control over it so that you can address the key questions in this paper? Nobody thought about anything else. Did you like this method? Were you convinced by the data?
I mean, I couldn't necessarily think of something better.
Yeah, so.
Yeah, I don't know. I guess the thing that sketches me out the most about it is just like how. I'm not really familiar with the methods they do. Like, I haven't done this myself, but I don't know like how that process affects the membrane integrity and like.
So that's an incredibly important question because lipids confuse.
Right.
You know, they can mix. The question is, what are the rate constants for all of that? And we don't really teach very much in the introductory courses about lipids and their partitioning between other membranes and fusion and all that stuff. But if you think about it, that's what the cell is, right? How do you get a plasma membrane and all these membranes around all these little organelles? That's sort of an amazing observation. And we've seen it in class already. What have we seen to get LDL receptor from here to the plasma membrane? How do we have to do that? We had to use these little vesicles, right? So you're generating something over here. It goes through the Golgi stack. Again, another set of membranes. It's got to come out the different levels of the Golgi stack, and then it's still got to get into the plasma membrane and fuse and dump its cargo. So I think it's sort of an amazing process, and people interested in evolution, this is one of the major things people are focused on, is how can you make cells, little fake cells, artificial cells that can replicate themselves. You can make it, and they're going to have to divide and fuse, and it's exactly the same problem here. And so this question of fluidity is an extremely important question, and a lot of people that focus on lipids, which is not a popular target, a popular thing to study because it's so hard, it's incredibly important. And people that look at membrane proteins, you know, they almost always have lipids on them, and then when you do them yourself, you have a detergent, which is not a real lipid. You know, does that change the properties? So all of these questions, I think, are really sort of central to what happens in the membranes, which is a lot of stuff inside the cell. So I think it's good to question what they did. I think their results turned out to be quite interesting, okay? But we'll come back. I think that was a hard problem. And so we'll come back and we'll look at this. And so then let's say that we could end up separating things. Then the question is, what was the key type of measurement they made where they could correlate the changes? They had to correlate the changes in cholesterol levels. We've talked about you can control perhaps the cholesterol levels with the cyclodextrin. But then how did they correlate the changes in the cholesterol levels in the membrane with this transcriptional regulation? Okay, and so that is what happens with the steroid responsive element binding protein, the transcription factor. So what happens in that process? What are the changes in the SREBP dependent on the concentrations of the cholesterol? And how did they take advantage of that in answering this question about what the cholesterol levels were that allowed you to turn on transcription of LDL receptor and HMG-CoA reductase. So what's the major assay? We'll look at that as well. So if you go back and you look at the model, what happens in this model? All right, here we go. What happens in this model? What's happening to SREBP?
There's a confirmation of change that expose the zip code region.
Now that's SCAP. SCAP, that's this guy. Okay, so SCAP, that's a key player. That's what we talked about. I know the names are all confusing. See, you're going to need to write these down to remember the names are very confusing. Yeah.
So the SCAP SREBP, like, I don't know, whatever you would call it, complex moves to the Golgi apparatus.
Right.
And then part of it's cleaved. And
Right.
moves to the
right? So how could you take advantage of that? This is the key observation that they're taking advantage of, to ask the question, since this whole process is dependent on the concentration of cholesterol. If you have high cholesterol, there's no way you want this to happen. You want to shut it off. If you have low cholesterol, you want to turn these guys on. Okay, so this movement is the key. And what do we see if we look at what happens to this protein, SREBP? What happens to it during this process? It gets cleaved, and what happens to, how could you monitor that cleavage? How do they do it in the paper?
They used was it a fluorophore, or is that the homework?
Well, they could use a fluorophore. They didn't do that.
That's the homework.
They did a what?
No.
They were able to separate the two SRIBP species just with a gel.
Yeah, so it can separate by a gel. So to me, this is actually quite an easy assay, because if you look at this, I don't remember what the molecular weight is, but it's a lot smaller over here. And so that turns out to be a great assay. So that part of their analysis, I think, was a really smart part of the analysis. And so then the question becomes, can you quantitate all of this? So if you have a lot of cholesterol, this doesn't happen, and so everything is bigger and resides in the membrane. You could even probably look at that. Whereas when the cholesterol is really lower, things go there, and it's everything in between. The question is, what is the concept? Can you measure, if you have X% cholesterol in the ER, how much do you have to decrease it to see a change or a switch in where this protein goes? So I think the experimental design is actually amazingly creative, okay? But then you see the data of the other side, and what I want to do now is sort of focus on what the issues are, okay? So we're going to come back and look at how did they look at SREBP. So you could look at this a number of ways. You could look at this by protein gel directly. How else do people look at proteins? Using Westerns. What's a Western? Anybody know what a Western analysis is? Didn't I ask you that at the beginning of class? How else do you detect proteins? You've seen this in the first half of the semester a lot.
Antibodies?
Yeah, antibodies. So if you have antibodies to this, and we'll talk about that, because the Western analysis, which people use all the time, and you can't, there are so many issues with it that I think I want you to think about what the issues are, okay? And then you correlate the two, changing the levels of cholesterol, which they measure by mass spec after separation and purification of lipids and the cleavage, and then they plot the data, and that's where they got the analysis from. Okay, so the first thing that you want to do, the first thing and the key to everything, is separation of the membranes. Okay, and so this is a cartoon of when you put something, you load something on the top and you have a gradient, and the gradient could be made of a number of things. Have any of you ever run these kinds of gradients? Okay, so you can make them out of glycerol, you can make them out of sucrose. Did anybody look at how these gradients were made? Did you read the experimental carefully enough to look at that?
The sucrose gradient.
Yeah, how do you make a sucrose gradient? You have no idea, but huh? Yeah, so layering. So what you'd really like to do is have a continuous gradient or something, but the sucrose is incredibly viscous. So if you were trying to make a linear gradient, which you could do by mixing two things of different concentrations, if you could get them to stir really well and then add it in, and you could generate a gradient. But it's so hard to do that what happens is they end up layering it. So they make X%, Y%, Z%. They put it down, okay, and then they try to layer something on top of it, and then they put whatever they're interested in at the top, and then they centrifuge it. Okay, so what are the issues? Do you think this is what the gradient would look like? So what are the issues when you're doing this, when you layer it? So and this is why the data, which we'll talk about in a minute, is the data. Part of the issue is this method. That's why you need to think about the method. And there are better ways to do this, and it really depends on what you're trying to separate. You know, so if this band, there was a little, say these were two bands, you wouldn't really get very much separation at all if there were two separate things that sedimented under these conditions very close together. Okay, so what would happen when you're sedimenting this? I mean, does anybody have any idea how long it takes? Do you think you do this in like a centrifuge, just spin it for 3 minutes and then, you know? So sometimes you sediment these things for 16, 20 hours. Okay, so what happens during the sedimentation that might make this more challenging in terms of separating what you want to separate? Yeah, so exactly, you have diffusion. And even when you've layered things on top of each other like that, you start to have diffusion. And if you shake up the tube a little bit, it's all over. So how you prepare these things is not. So people still use these methods, but I would like to see better methods. And so they tried, you know, one method with sucrose, and then that wasn't good enough. We'll look at the data. So they went to a second method. And, you know, where did they come up with this? I have no idea where they came up with this, but there's an MD PhD student in our class who has seen this in one of his classes, and they use it in some blood test. So I think that's probably where these guys got it from, because they're both Brown and Goldstein are both MDs. But again, it's just another way to make a gradient. And I'm not sure why this gradient works as effectively as it does. But the first gradient didn't work so great, and we'll look at that data. So then they added on a few more steps because they weren't happy with the level of separation. So looking at membranes, I mean, I think this is going to be more and more looking at membranes, because membranes, you have two leaflets. The lipids in the leaflets are different. Do you think that affects the biology? I guarantee you it affects the biology in ways that we would really like to understand that I don't think we understand very well. When you isolate a membrane protein, have any of you ever isolated a membrane protein? No. So you have an insoluble, you know, it's in this lipid system. How do you think you get it out so you can go through the steps that, of protein purification that you've talked about or have probably done in an introductory lab course? What is the first thing you need to do? Yeah, solubilize it. And how do you solubilize it? Yeah, with some kind of detergent. It's sort of like what you saw with the chylomicrons, right? Or the bile acids that we talked about. So you can use different, and people have their own favorite detergents. But again, that changes things. But otherwise, you can't purify anything unless you happen to have a membrane where, you know, the only protein in the membrane is the one you're interested in, which of course doesn't exist. Okay. So anyhow, they went through that, and then what did they end up seeing? So they went through different steps, and they separate them into different, the supernate and the, or the light and the heavy membrane fractions, and then they have to analyze it. And so the question is, how do they analyze to tell how well these separations actually worked? What was the method that they did to determine whether they separated the ER from the plasma membrane, from the Golgi stacks, from the lysosomes, from the perioxysomes? Okay, so we have all these little organelles in there. What did they do to test each one of these fractions? Okay, let me ask you this question: how do you think they got the information? How did they get the material out of these gradients to do the experiments I was just talking about? So they want to analyze what's in each of these bands, okay? How do they get it out of this tube?
They use a Pasteur filter.
So what do you think? You just stick it down in and suck it out? Well, I mean, you know, so what do you think? Say you could do that, right? You open the top, you stick it in, you carefully stick it in. If you can see it, lots of times you can see these lipids because they're like opaque or something, so you can see, or if you have if you still have your sucrose layers, lots of times they layer in between the different concentrations of the sucrose, and you see white stuff precipitating. So you could conceivably stick a pipette from the top and suck it out. Okay.
Perturb all the other layers.
Absolutely would perturb all the other layers. So here you're doing something. It's already a very hard experiment because they're all being perturbed anyhow because of diffusion. So is there any other way you could think about separating these things? And so the hint is that they use plastic tubes. So these things are not glass, you know, no centrifuge.
Freeze it, cut it.
Well, I mean, no, so you don't do that. That could be, yeah, okay, so you could, but then, but you then have to, if you were cutting it, you'd still have to get it out of the tube, right? Okay, unless you had a saw that didn't have any vibrations when you were cutting it, of course, which would not happen. Okay, but if you look here, in this cartoon, so I gave you this, what are they doing here? They're sticking a syringe in through the side of the tube, and that's still what people use. So you can suck out, if you can see something, you know, so you have to be able to see it in some way to know where to suck it out. So you might have a way, actually, in doing ultracentrifugations, I think with the lipids you can see them by eyeball, but you might look at absorption. If they have proteins, you could monitor absorption through the gradient, and that might tell you how to fractionate things. But anyhow, that's also an issue, because before they can do the next step in the analysis, they've got to get the material out. Okay, so they've got the material out in each of these steps, and then how do they look at this? They can pull it out. So what are they looking for? To tell them how effective this method is.
They look at some specific markers for each
Right,
problem.
exactly. So what are they, so to do that, what they're going to have to do is, before we look at the details of the method, I want to go through a Western blot. Okay, so what do we know about a Western blot?
I have a quick question on
Yeah.
the lysis method.
Yep.
There. It says
About the which method?
the lysis method.
Yeah.
The ball bearing homogenizer.
Yeah.
So they're literally putting these cells in something with a bunch of ball bearings.
Yeah, you could do that. Yeah, I mean, there's a lot of ways to crack open cells. I don't know which one's the best. Mammalian cells are really easy to open. Sometimes all you have to do is freeze and thaw them. Sometimes you have like a little, just a mortar and pestle or something like that. But I mean, yeast cells, you roll them. You have to have enough cells so you can do something. If you only have a tiny amount of cells, it makes it really challenging with beads because it covers the beads. So you,
do you have any issues with any of the different types of membranes that?
Sticking to that, absolutely. I'm sure you have to look at all of that kind of stuff. So how you choose, that's an important thing to look at, how you choose to crack open the cells. And I mean, it's the same with bacterial cells. There are 3 or 4 ways to crack open the cells, and I can tell you only one of them really works efficiently. And a lot of people, when they use some of the others, you know, they do something and they assume it works, but they never check to see whether the cell walls have been cracked open. A lot of times they haven't, and so what you get out is very, very low levels of protein because you haven't cracked open the cells. So figuring out, I mean, mammalian cells are apparently, I haven't worked with those myself, but they're apparently much easier to disrupt than bacteria. Or, you know, if you look at fungi are really hard to crack open. Yeast or, so anyhow, that's an important thing to look at. So every one of these things, I mean, again, maybe the devil is in the details, but when you're doing your own research, it doesn't matter what method you're looking at. The first time around, you need to look at it in detail and convince yourself that this is a good way to chase this down. And you look at it in detail the first time around, and when you convince yourself it's working really well and doing what you want to do, then you just use it. And that's the end of it. You don't have to go back and keep thinking about this over and over again. But so the method we're going to use is a Western blot. Okay, so we got this stuff out, and have you all run SDS page shells? Okay, so SDS page shells separate proteins how?
Based on size.
By the what?
Oh, it separates into like a charge gradient and then not charge gradient, but
Not charge.
what drives protein, but it's based
Right,
on
but
size.
you have, yeah, it's based on size because it's coated with, every protein ratio is coated with this detergent, sodium dodecyl sulfate, which makes them migrate pretty much like the molecular weight, but if you've done these, it's not exactly like the molecular weight. You can do standards where you know the molecular weight, you can do a standard curve, and then you see where your protein migrates, and sometimes they migrate a little faster, sometimes a little slower, but it's okay. So you run this, and then what do? Does anybody know what you do next to do a Western?
You need to use a membrane to.
Right, so the next thing they did was they used, whoops, I'm going to put all of these up. So they transferred it to a membrane, okay, and why did they have to transfer it to a membrane to do this analysis? I mean, this is an extra step, and it turns out, so, you know, we're going to look at an antibody interacting with a protein. Why don't we just look at the antibody interacting with the protein to start with?
It wouldn't have access to the
Right, it doesn't have very good access. It's really not very efficient. So people found, pretty much by trial and error, that you needed to transfer this to a membrane. I mean, we have hundreds of kinds of membranes. How did they choose nitrocellulose? Okay, if any of you have run Westerns, do you know, remember what kind of a membrane you used? Did you use nitrocellulose? You do this in undergraduate class, don't you? You don't do a Western? Okay.
Yeah,
We used to do,
did once in undergrad class.
yeah, in what kind of a membrane? Was it in biology?
Yes, about.
Okay. So what membrane, do you remember what the membrane was?
It's not natural cellulose.
Yeah, it's not nitrocellulose. So this PVDF, polyvinyl difluoride, is the standard one that people use now. It works much better than nitrocellulose. This paper is really old, and so they're looking at nitrocellulose. Okay. So then they do this, okay, and then what do they do next? They have an antibody. We'll look at the details of this in a minute, that can recognize the protein, that can find it on the membrane, okay? And then what we're going to see is you still can't see anything, really, because you don't have very much material there, and you can't observe it. You don't have enough to stain, oftentimes, by Kumasi, so you're going to have to amplify the signal. So then you're going to make an antibody to an antibody, and then you have to figure out how to then amplify the signal, and we'll look at that in a second. Is this what, you ran a Western, is this what Westerns look like?
I remember we first flow non-specific proteins to occupy the size of
Yeah, so that's good. You have to block everything if you're using crude extract. Okay, so in this case, we would be using the crude mixture. Well, not a crude mixture. It's been fractionated by the ultracentrifugation. It's been fractionated, but you still have mixtures of proteins in there. Have any of you ever looked at Westerns in a paper? Well, even the papers you had to read. The paper on the PC, go back and look at the PCSK9 paper, that had Westerns in it. What do you see? Do people show you something that looks like this? And if they did show you that, what would it look like? So you have an antibody that's specific for the protein of interest, whatever that is, supposedly specific. What do you see? What do you think you see? Do you think antibodies are specific? I think I have an example of a typical Western.
I don't think they're as specific as they make them look.
Yeah. So when you look at a paper, you should go back. You should pay attention to this when you read a paper. If you're doing anything in biology, what do you see? You never see a gel, ever. What you see is a slice of a gel, where they cut off this whole. Where they cut off all this stuff and all this stuff. The reason they do that is because it's a hell of a mess. Okay. So let me just show you a typical. I mean, I don't care what kind of a antibody you're using. In crude extracts, it's a mess because you have non-specific interactions. We'll just look at that. So that would be something like you might see depending on how much antibody you have. So when you see this, the reason everybody reports data like that now. So it looks like it's really clean, but in reality, I think if it's as dirty as that, then in my opinion, I would make you publish the whole gel. But people don't do that. They just cut off the little band they're interested in. They can see it change in concentration using this method. But you should be aware of the fact that antibodies in general aren't as specific as you think they're going to be. Yeah.
Are they required to report like the full gel in like supplementals?
Well, I mean, I think probably depends on the journal and it probably depends on the reviewer. But I would say we're going away from data. This is something that is a pet peeve for me. We don't, and all the data, which I think is all right, is published in supplementary information as opposed to the paper. I think if you have something really dirty, you should publish in the paper, in the main body of the paper. If you have something that's really clean and it looks like that, it's fine with me. You don't even have to publish it if you could believe what people were saying, okay? Because people know what this looks like. A lot of people, everybody uses Westerns. But if it's a real mess, then you need to let your reader know that this is not such an easy experiment and it's not so clear-cut. That's what your objective is to show people the data from which you drew your conclusions, and then they can draw their own conclusions, which may be different, okay? So let's look at the apparatus to do this. Okay, so how do you get from here to here? So you have a gel, you run the gel, a polyacrylamide gel. What do you do?
The membrane on the gel.
So you put the membrane on the gel, and what do you do? Okay.
You apply charges to.
Yeah, so you're transferring it based on applying a voltage across this system. So here's your gel, okay, and here's your membrane, nitrocellulose membrane, okay, and then they have filter paper above the gel and below the membrane. Why do you think they have the filter paper there? When you ran the gel, did you have filter paper?
Yes.
Yeah. How do you think they decide how to do this transfer? Do you think this is straightforward? Do you run it for an hour? Do you run it for 5 hours? Do you run it for 15 minutes? What is the voltage you use to do the transfer? Do you think any of that is hard to figure out? Okay, so how do you figure that out? Somebody told you that this is a good way to do it? Yeah, so that might be a place you start, okay? So you do it because somebody gave you a recipe, okay? But then what do you need to do to make sure this recipe is correct?
With conditions that work for what to work.
Right, so, and then how do you do that? So that's true. Every protein is going to be different. And if you have a protein, if you have a clean protein versus a mess of proteins and you try to do this transfer, the transfer conditions will be different. So, for example, if you really want to look at the concentration of something inside the cell in the crude extracts, you never compare it to a standard with clean protein because this transfer is different. So you need, in the back of your mind, if you care about quantitating this, you need to understand the basis of the transfer. Okay, so why do you think they have these filter papers here? So this goes back to what controls you would do to see whether your transfer was working. So what would you look for? Did you do this?
Yes.
What did you do? Why did you have what did you do with the filter papers in your?
Filter out the SDS molecules.
You did what?
You want to filter out.
No, that's not what you do. I mean, you might want to do some of that too, but what is in terms of thinking about whether your transfer is successful, figuring out the conditions to blot from the gel to a piece of paper is not trivial, okay? And so there is a standard way that you do this initially to try, but then you have to make sure that that method is working, and lots of times it doesn't work. So it's something that's going to be experimentally determined. So the question is, what happens if you did, what would you think would happen if you did this for 6 or 7 hours, whereas a normal blot would take 2 hours?
Would you transfer tons to the filter paper?
Right, it would go right into the filter paper or even off the filter paper. So what you do is you take the filter paper out and you look for protein being bound, okay? What about the gel? What do you do with the gel after your experiment's over?
Make sure there's protein not on it.
Right, make sure that the protein's not on it. So these are simple controls, but these are the controls you always do until you work out the conditions to make sure this works, and it's pretty critical to make sure you have good transfer. So then if you look at their, so this is the antibody thing that they do. Has anybody thought about these kinds of assays? You've seen them, I think, already in class, but you have, what's wrong with this picture? The target protein. What's wrong with this picture of the target? So here's your nitrocellulose filter paper. What's wrong with this cartoon? Should be unfolded, yeah. So you're doing STS page, it's unfolded. So then we react it with an antibody. Presumably we have a good antibody, but you've already learned in the first half of this course that having really good antibodies is not so trivial. You can get them, but most of the time they are not specific. If you're looking at crude extracts, they have little epitopes they recognize if you're using monoclonals that could be present in other proteins. And furthermore, how are you detecting something? An antibody is a protein. It has absorption at 280. Again, this is too low to see. So putting an antibody on it is still going to be too low to detect. So how do you detect your signal? So have you done this? I'm surprised they don't do this in your introductory class. They don't do Westerns at all, okay? So what you're looking at is an antibody to an antibody. Okay, so you put your antibody on, that's specific for your protein, and then you make an antibody in another organism that can specifically recognize antibodies in general, okay? So if this is to a mouse, you make it in a goat and isolate that. And then what you do is derivatize the second antibody with what? A protein, okay, that can function as a catalyst.
Why can't you just derivatize the first antibody?
Well,
Okay.
then what? What did you say?
It's more expensive?
Well, no, I mean, I don't know whether it's more expensive or not, but
Well, because you'd have to derivatize every primary antibody.
yeah, so you'd have to derivatize every primary antibody, and so this is sort of a standard procedure. You could derivatize the primary antibody, so that's not a bad question. And so what you're doing now, you can buy these commercially, you know, so they have goat, rabbit, mouse, whatever, antibodies. And the key is the amplification of the signal, and you use enzymes to amplify the signal. Does anybody know what the enzymes are? What the enzymes do to amplify the signal?
Convert a molecule to a blue molecule, I think.
Yeah, to something that's colored. Okay, so does anybody know what that horseradish peroxidase? Have you ever heard of horseradish peroxidase? So that's a heme iron. We're going to be talking about heme irons pretty soon, and hydrogen peroxide. It makes a chemically very reactive iron oxo species that can oxidize a dye that changes color, and it has an extremely high extinction coefficient. So you can see it, and it does it catalytically, and the lifetime of the dye is long enough so it accumulates, and you can get really amplification of your signal, or you can use a phosphatase that liberates something that's highly colored, again, and you can see it. So this is a standard method that everybody uses. And so that's our gel. So now we're looking at sort of, we're at the end already, but we're looking at these gels, and what do you see through the different steps? So if we look through the first gradient, through the sucrose gradient, that gets us through D and E, okay? And if you look, say, at lane E, and our goal is to separate proteins that are specifically localized in each one of these membranes. So you need to believe that's true, okay, that people have selected the right group of proteins to look for, and you notice they do more than one, okay, so they look at multiple proteins. Why do you think you need, do you think it's easy to select the proteins to look for? And why or why not? So, I mean, they obviously have selected a group of proteins, and I think, you know, most people would agree that they've selected a good group of proteins. But what do we know now about proteins? Do they stay in one place? You know, they move around, and but someone might be present in very low amounts, sometimes in much higher amounts, and so you need to have more than one protein as a control to make sure you're looking at in the right region. And what do you see in E? If you look over here, it tells you what the organelle is, and if you look at this protein, this is localized to the lysosomes. We talked about that in class. If you look at this protein, it's localized to the peroxisomes. So in addition to the ones we care about, the ER proteins, we're also getting proteins that are localized in other membranes. So that's when they went to the next method, and they added on another gradient to try to separate out, again, the lysosomal and the peroxisomal proteins. And you can see they were pretty successful at this. There's none of these proteins left in this gradient. Okay, so that's good. And they took it a step further. Do you remember what this is? What are they looking for down here in this?
Enzymatic activity.
Yeah, so enzymatic activities are localized in certain organelles. So they, again, did a second experiment to look at all of that. So, I mean, they were very careful in this. They figured out how to separate, and that's the key thing for them to analyzing the concentration of cholesterol in these membranes. And what they looked at, we're over time, but is the concentration of cholesterol compared to the total amount of lipids, okay? And how did they do that analysis? You know, Gene Kennedy, who's at Harvard Medical School, I think he's in his 90s now, really trained all the lipid chemists in the whole country. And they figured out many years ago how to separate lipid fractions with methanol, chloroform extract, something that you guys probably haven't thought about at all. But we're really pretty good at separating things, and it's nothing more than an extraction like you do as organic chemists to purify and separate things. We figured that out. And so then they use mass spec to allow them to quantitate the amount of glycerol. And then in the end, so they use mass spec, they use Western blots, and they can change the concentration of the cholesterol and do the experiments over and over again to see what happens. And when they do that, this is the picture of the cyclodextrin. So you can see the only difference is this group here versus that with a methyl. And so this is hydroxypropanol, cyclodextrin. Okay, so it's like a cavity like this. And only other change here is a methyl group, removing that. And they have very different properties about binding and releasing cholesterol, which somebody had to do a lot of studying on to be able to ensure that they could use it to remove cholesterol and then to add it back to the media. And so you have to think about the exchange kinetics. You have to think about a lot of things. This is not trivial to set this up to figure out how to control the levels of cholesterol. And then what they do is, this is like a typical assay, and this is the N. What you can do is this removes cholesterol, and you can see it change. This reports on low levels of cholesterol, which is happening over here, allows the protein to move to the nucleus where it's smaller, and that's how they do the correlation. The correlation between the levels in the nucleus and the levels of cholesterol. So I thought this was a pretty cool paper, and these kinds of methods I think will be applicable to a wide range of things if people ever do biochemistry looking at the function of membranes. So, okay, guys.